So from what I understood, it's the crowd has some people interested in math, some other in biology, some in bioinformatics. So I will try to feed a bit for everyone, which is not an easy task. But there are still questions that we are all interested in population genetics. So maybe I'll start first asking the questions and then see if we can say something about it. So, so the topic of this presentation, of this talk, is to explain the processes that are causing the genetic diversity we observe within species. So, I chose the picture from the first slide, not randomly. First of all, I guess you can see my mouse pointer.
Yes.
Okay. So first, there is a picture of Don Quixote, like hunting for giants that are not giants. So it refers to the idea of like we have something in front of our eyes with our own way of seeing those things, but we don't really know what they are, right? We can only analyze them through the lens of our models and of our way to see the world. And I kind of reach a point where I start like bugging myself and like trying to say, what do we do something that makes sense, or we do something that doesn't make sense anymore, right? And I think it relates to the genetic diversity problem. That's an old riddle of population genetics, and it can be recast as a time frame problem. So we'll see that it's all about time scale. I think that's the key to get out of the riddle. Okay. So just a very broad sketch of the way I would like to think about the process. So on the right side, we have the individuals here. And that's the observed patterns. And here I chose humans, but I'm not focused especially in humans. So there is a collection of diversity. Some individuals are like, say, more gray, some other more black, some are greens and red and orange. And we can classify the individuals and saying, for example, the red and the orange, they more look alike, or the one without the colors, they form a cluster. And likely they form clusters because there is a pattern, there is a kinship tree behind those individuals. And on one side, going from left to right, from the kinship tree, we can go to the pattern of diversity. That's probably what happened in the biological side. And if we go on the other way around, if we go from the individuals to the kinship tree, that's like the phylogenetic approach. Like, can we find the tree that relates individuals, provided that they're in the same species or not? It changes the question a bit, but it's the same kind of approach. But here we are even more ambitious. We would like to understand what are the processes that create the tree. So on the left side, we have like some kind of a branching process where the lineages, they branch and they die, and they form a population that is now expanding in that example. And what we do have at present day, we have a sample of individuals that are the yellow disc here, the small discs here, and those individuals, they are related by the process through a kinship tree, and we have the observed data. So there is a game of like the biology goes from the process to the individuals, and there is a game we want to go from the individuals to the processes. Okay, so that's the general framework. So a very quick, rapid tour of ancient history in molecular evolution. So I think important dates and people are the following. So an important observation is in 1962, where Emile Zuckerkandel and Linus Pauling noticed that when we count the number of genetic differences between two individuals belonging to two different species, somehow there is a linear relationship between the number of mutations that have occurred in the two different lineages that went to the two different individuals belonging to the two different species. So the individual, they kind of accumulate mutations linearly with time, at least at the between species level. Another important date is the first analysis of polymorphism by Lewontin and Hubby in 1966 that show that surprisingly, if you look at any locus, at any species, you'll find some polymorphism, which means there is diversity among individuals within the same species, in all species, in all individuals, in all samples, and in almost all loci. Built on those two observations, was proposed that the patterns of polymorphism that we see, the polymorphism that segregates within the population and then accumulate between the species, are best explained with what we call a standard neutral model. So that was proposed by Kimura in '68 and popularized later on, culminating with the book in '83. Right, but very soon after the proposition of the neutrality as a good explanation for explaining genetic diversity, there was already someone like Richard Leontin, who passed away very recently, who already pointed something strange in the theory. That is, under the neutral theory, the diversity within the species should scale with the population size, right? And apparently, he pointed out there is no relationship between the population size of the species, the number of individuals within a species, and the diversity within a species. That's only the beginning of a very long debate that is still very full of emotion and still occurring very recently, and I put a recent paper like attacking strongly the standard neutral theory. Okay, that's the history slide. So what's the standard neutral model? So the standard neutral model is the model assuming that when you model a population, you have to assume that the population size is constant. And that's my gray tube here. And as time passes by, the population size doesn't change. The population size, the number, the mixing between the individuals is full, say complete. It's a strict parmixia, and there is no selection, right? So one of the consequences of that is the number of descendants of each individual is a random variable with a mean one, and it's a Poisson distributed. Everyone has the same chance at every generation to leave descendants, right? You can have a haploid version, diploid version with the sexes, and it doesn't really change the major conclusions. So two classical implementation of that standard neutral model is the so-called Wright-Fischer model. So I assume everyone is more or less familiar with that, so I'm gonna be very quick on that. So in a Wright-Fischer model, here you have time, and generations are discrete. At every generation, everyone dies and is replaced by a random sample of the previous generation. So for example, that individual is a descendant from this one, these two descend from this one. And this is a real realization of the process. What we notice when we do that is, in the long run, only a single individual that I have highlighted here in black will be the descendant of the whole population in the future. I mean, everyone else will have a descendant that will vanish at some point in the future. So there is a single lineage of ancestor in that model, and because there is a single lineage of ancestor, in the long run, only the mutation that occur in that lineage will persist, and that's a good explanation for the molecular clock. There is a single lineages, and only the mutation that fall into that lineages will persist and accumulate between the species. Okay, so that's one way of implementing the standard neutral model. Another way is more recent by Patrick Moran in the 50s, where now we have time steps and the generations are completely overlapping. At each time step, that could be a random time or fixed time, you pick an individual to reproduce, and here this is this one, and an individual to die, and here this is this one, right? So see, the number of individuals remains constant. One is giving birth, one is dying, and you repeat again and again and again this process. So in both models, there is no beginning or end, right? There is still just repeating the replacement of the individuals. And this creates this replacement, and this will create what we call genetic drift. So genetic drift is the invasion of a population by a progeny of someone. So say in the right Fischer model or in the Moran model, the line of ancestor is the black thick lines. So you pick any individual from the black thick lines and follow their descendants forward in time. So this is what I've done here. So I've picked that individual and I've colored all their descendants. And you see they slowly like invade the population up to a point where they reach 100% of the individual. And corresponding to that, you can look at the frequency of these progeny, and it goes up and down like drifting in the space, hence the name of genetic drift, right? Another way of looking at the same process is to look at time but backward. So instead of picking an individual and looking at the progeny, you sample the whole population. Here I've sampled the whole population, say here, at the last time steps. So I colored every individual in black here, and I followed their ancestry, right? So what I did is I colored their ancestral lineages up to the point where they reach a common ancestor that is colored here, right? So you can look at this process either like forward, that I will call the genetic drift, or backward, that I will call the coalescent process. So coalescent means finding some common ancestor or looking at the process backward. Okay. So interestingly, you never specify the tree in that process. You just specify the reproduction of the individuals, and you see there is a tree of kinship relating all the individuals emerging from that process. So there is interesting properties of like how do we relate the forward and the backward process, that is called the duality. But that's not the topic I'd like to talk today. This is a fascinating topic. I'm gonna get lost if I continue on that topic. So just to summarize, the mutation drift paradigm, the standard neutral model, has taken the place of a real, say, statistical null model. So I will call it H0. And that model is simply, you have a pool of individuals. What removes diversity from that pool is the so-called genetic drift, that you can see forward or backward, like seeing the coalescent or the forward process. And it takes about capital N generations to remove diversity. But what creates diversity is simply that every single individual can mutate with a certain probability mu. And at diversity, at the equilibrium, we have an expected diversity of n times mu on the order of n times mu, right? So under this model, whatever is your favorite measure of genetic diversity, the number of polymorphic size, the pairwise difference, the number of haplotypes, whatever is your favorite measure of diversity, you end up having a function of n times mu. That is the population size times the mutation rate. Higher diversity is either high mutation rate or high population size. The question I'd like to discuss now is this the right model? I mean, we assume this model since the 70s as some kind of a reality, but is this really a reality, or is this just a model we like to do calculations on. So we'll be challenging the model. That's the idea. I will be challenging the model using two cases, two sub-topics. The first one is on the frequencies of the variance we observe in the population. And the second one is about the total amount of diversity. Okay, so let's move to the first case.
Sorry, a question. What is the S and M in the last slide? Yeah, this one for all S and M.
Oh, standard neutral model.
Oh, okay. Okay, thank you. And also
yeah. Oh, so please, I don't see the questions where I have the full screen. So please jump in and
no,
ask the question.
Okay, I just ask it audibly.
Yeah, it became my friend, so I had chosen a nickname for it, and now it's S&M. But so
okay, thank you.
So it's just it's a general class of model. Again, the way exactly you specify how many individual dyes and are produced at each generation doesn't really matter, right? Okay.
Okay, and also the diversity is O and mu, so this is order of magnitude in the unit of the product of n times mu. Sorry, I don't get this time scale, really.
Yeah, no problem. So if you measure any kind of metrics of diversity, say for example, you count the number of sites that are polymorphic in your genome or in your sample of genomes, the expected number of polymorphic sites, which is a measure of diversity, right, will be a function of n mu. It will be n mu times some constant.
Okay, so it's the order of magnitude is based on the number of the polymorphisms that we have from the mutation.
But that's only one metric of diversity. Another one is like you count the number of differences between two individuals. That's another measure of diversity. The average difference between two individuals. That's also functions of n mu. If you count the number of different genomes in a locus, different haplotypes in a locus, that's also a function of n mu. Whatever is the exact metric, you'll end up always having that measure of n mu.
I see the point. Okay. Thanks.
Yeah, great. Thank you for the question.
Thank you.
Okay. So that's the case one. So that's a manuscript we've been working for quite a number of years, but it's ripe for putting in bioRxiv. Just need to have a last quick read about it. So the manuscript is entitled "Interpreting the Pervasive Observation of U-shaped side frequency spectra," and it's a joint work with many colleagues. Some from Vienna, with mathematicians, bioinformaticians, biologists. Okay. So I have to make very clear now what recombination does the population level. So on the left, you have a pedigree with grandpa and grandma and their daughter that is happily married with someone else who had an infant. Okay. What we are defining as biologists as recombination is usually we see recombinations as like putting together some bits of genomes. So say here the daughter, she received half of her genome from grandpa, the blue part, and half from grandma, the red part. And during the meiosis, there was a recombination event. So the chromosome that she transmitted to her infant is a mix between blue and red. Okay. If we look at the process not forward but backward, what we do see is when we get back to grandma and grandpa, the blue part of the genome of this infant is carried by a different individual than the red part of this individual. Which means that the genealogy, if we trace back the genealogy of the genomes for the red part and the blue part, they will have somehow common genealogy up to the point where there was a recombination. Say, for example, look at the middle figure. So you have the genealogy, so you have sampled 3 genomes that are here half blue and half red. You look at their genealogy, they have a common genealogy, but here you had a recombination event so that the red bit of the genome has a different genealogy than the blue part of the genome, right? So anytime you find a recombination event in the ancestral lineages, you have a different genealogy. And at the genome scale, what it does is like create many trees that are known as a tree sequence, a very large number of trees, and depending on the chunk of DNA you're looking at, the genealogy will be different.
One nice question. This like
Yes,
phylogenetic tree is like a gene tree, or it would be like an individual tree? It would be like a gene tree, right?
Can you restate the question?
Yeah. If this phylogenetic tree is a gene tree, each node would represent like a different gene, for example, or a different locus from a gene.
So that's the genealogy.
Like it's also what it's representing, because I don't know what's each end of the tree is representing exactly.
Okay, so you've sampled 3 individuals. That is this one, and this one. And you're trying to represent their kinship tree, right? How do they relate one to the other? So depending if you look at the blue part of their genome or the red part of the genome, their genealogy will not be the same.
Okay, I think it makes sense. Thank you very much.
So, and this is the case where you have only two genealogies, but if you move further along the chromosome, there will be another recombination and another genealogy again and again and again. So there will be a very large number of trees, yeah, at genome scale.
Thank you very much.
So what does the recombination? It actually resample many trees, and what you could do is when you do genome-wide metrics, what you do is you actually averaging across all possible trees of the genome. So when you look at metrics at the genome scale, you're approaching averages across the tree space. And one interesting object that was developed in the population genetics is the site frequency spectrum. So that's the distributions of the frequencies of the mutation. So in the first bin here, you have all the mutations that have small frequencies, say 1 over your sample size. And since Foo 95, we do know that the expected site frequency spectrum is proportional to 1 over the frequency of the variant. So it has like a 1 over x function. So if you represent the site frequency spectrum, it should decrease as 1 over x under if the standard neutral model is true. Okay, if the standard neutral model is not correct, this won't be correct anymore. So one way to test for the standard neutral model is to look at the distribution of the frequencies of the mutation. So you collect all the variants in your alignment and you compute their distributions of frequency. So this is what I've done with like Eurobase. So Eurobase went through the 1000 genome project, get the 108 Eurobase individual. So that's 216 haploid genomes. That's about 80 million variants. And simply count their frequency and do the distribution. And what you do observe is the diamonds here, which means, well, there are more rare variants, and then the very frequent variants, there seems to be less represented. Note that in that representation, I did not take into account the R groups, so I don't know if a variant is very rare or very frequent. So what we do is we fold the spectrum and put together in the same bin the very rare variant and the very frequent variant. So this is what you see, which means you don't see anything, right? And what you expect from theory under the standard neutral model is the red line. So it looks like it's not that bad, but is it the correct model or not? Difficult to say visually, right? So what I've advocated for a number of years is like to use a transform version of the spectrum where you divide the observed value here by the expected value under the standard neutral model. If the standard neutral model is correct, you would expect this red line. And using that transform version, you immediately see that there is an excess of low frequency variant. That means the variants with low frequency, they are overrepresented. And the variants with a medium frequency, say around 0.5, they are underrepresented. So you have to explain that the standard neutral model is not predicting enough rare variants. So what you do what we like to do in human population genetics, you put a demographical model, and here I've fitted an exponential growth, and see with an exponential growth of parameter 0.7, I can reproduce more or less the observed side frequency spectrum, which means, without any surprise, the human population is expanding, maybe exponentially, maybe not. We've shown in the past that actually any kind of growth will explain the data for the side frequency spectrum. So okay, that's the typical way we do things. We have a standard neutral model, we add demography, sometimes we add structures, and that's it. And we fit the data, and we're quite happy with that. So what we've played with is unfolding the spectrum. So unfolding the spectrums means you use an outgroup to orientate the derived mutations and the ancestral mutations. So when you do that, you have a spectrum that go from so the mutation that could be carried by one individual up to 2015. Okay, everyone but one individual, and you have that spectrum, and again you don't see anything, right? But what you could do is use the same trick. You divide the observed spectrum, but what you expect under the standard neutral model. And now, with great surprise, what you see that there is still a bit of excess of very rare frequency variant, but there is also a very strong excess of high frequency variants. So now your problem completely changed, right? You had to explain that why we had an excess of low frequency variant in the previous representation. And now you have to explain why do I have an excess of low frequency variant and very high frequency variant. And if you look at demography, say you put growth, what you could do at best is the blue line, which is an extremely poor fit. And in general, if you think about non-extreme demography, like a demography jumping from very large size to very small size in a very short time, you cannot generate this excess of high frequency variant. But what you could suspect is that you made an error while using the outgroup. Maybe what you declared as a derived variant is actually an ancestral variant and vice versa, right? Because you use an outgroup for that, and it has many good properties, but there is a risk that actually you confused ancestral and derived for a given variant. You think that mutation is derived, but that variant is actually the ancestral version. So what you could add in the model is the probability of swapping between the ancestral and the derived variant. And you can even estimate that probability using the data, right? So you have a model that have an exponential growth and a probability of mixing ancestral and derived variants. And using some classical statistical techniques, least square or maximum likelihood, you can estimate the parameters, and what you'll find is the best model you can do using the Yoruba population is estimate some kind of growth, slightly larger, and 2% of error, which means when you have orientated the variant, you have a 2% chance that you made an error. So you end up with that green curve, which is better, but still see, I was not entirely satisfied by the fit. In the middle, I found that the green line, the model, did fit better, clearly, but did not fit so well with the data. So we figured we could do, as Monty Python suggested 50 years ago, something completely different. So we've been playing for all these years with what we call the multiple merger coalescent. So the multiple merger coalescent is the class of kinship trees where you can have polytomies. So polytomies are that kind of node here, where a node has multiple descendants. Under the standard neutral model, the kinship tree, the tree that relates all the individuals within your population, has only bifurcations, right? So it's a bifurcating tree, and it's known as the Kingman tree, named after the discoverer of this tree, of the distribution of this tree. But you know, when you look at like the trees between species, we do know there are radiations, like nodes with many descendants. If you look at the radiation of the mammals, they are all like originate almost as a single time point in time, well, at least at that time scale. So in population genetics, people have worked on what they call the multiple merger coalescent. Among the multiple merger coalescent, that is a very large family of trees, there is a family called the lambda coalescent that has been described in just before the 2000, where the lineages went. So you look at the individuals and you look at their tree, and there is a small chance that you select few individuals and you put them into a single ancestor, that you do a multiple merging event. Right? So there is a whole mathematical framework for that, based on these rates of transition. That's the probability that when you have B lineages, B individuals, you will collapse K of them at a single time point. Right? And there are subversions of this. One is called the psi-coalescent, and it has been proposed by Bjarke, Eldon, and John Wakely like 15 years ago. So this model corresponds to a case where you have a standard neutral model, but at sometimes in the population, you have a large genitor that will have many descendants. Say, if you go back in history, you'll find some kind of a Genghis Khan every that much generation that will have many descendants, right? So that will cover a macroscopic fraction of the descendants. So that's one way to specify the multiple merger. Another one is to inflate the variance of each individual, of the progeny of each individual in your population. That is, in the standard neutral model, I told you the distribution of the number of descendants per individual is Poisson of mean 1, and here you have another distribution with a larger variance. So some individuals will have many descendants in some generations. So that's the same idea. You inflate the variance of the number of descendants. And the mathematical apparatus is it's quite different between the psi-coalescent and beta-coalescent. And the both models are interesting because they have a parameter. In the psi-coalescent, the parameter is psi, and in the beta-coalescent, the parameter is entitled alpha. And playing with psi or alpha, you can interpolate between trees that are complete radiation. Say here, a beta-coalescent with alpha of 0 or a psi-coalescent of psi 1 is a radiation tree. So you have a single ancestor, and all your lineages are common descent of that ancestor. up to a Kingman tree that is a standard neutral model. So now you have suddenly a new parameter you can tweak and play with and see if you can fit the data. So we've been implementing that MMC model. So just doing the MMC, so that's the beta model with alpha of 1.9 almost, which is smaller than 2. And remember, 2 is the Kingman coefficient. So allowing for an alpha that is not 2 give rise to the fit of the purple one here. And if you even allow for errors, you will have that fit in orange, which seems slightly better from this one, from the green one here, right? So by playing with that parameter, maybe you gain an explanation, and you can even add demography onto that. And what you could do and see in a likelihood framework, what is the gain of likelihood of adding like orientation errors, growth, so demography, and these multiple merger components. So see, that's a gain in log likelihood. So I put the reference model here, the vanilla standard neutral model, so no demography, no error for here at 0, and I compute the gain in likelihood and see if you add either error or growth, you gain something. If you had growth and error, that was the fit I had before, you gain a lot in log likelihood. But anytime you allow the multiple merger component to be different from the Kingman, there is a big gain in likelihood, right? And obviously the best model is like growth, so demography, some multiple merger component, and some errors. And statistically, you would choose the model with the 3 parameters for the Euro. So even for the humans, what you would choose is the model with some larger variance for the number of descendants. Okay. So now we've studied not a single species, but we've collected 45 species with some bacteria, some plants, some invertebrates, some vertebrates. So the deal was to have at least 10 individuals and being genome-wide SFS. So I removed any kind of data set where there was a single locus or say 10-ish locus. Right? I really wanted to have a genome-wide polymorphisms. So you can do a visual representation of the site frequency spectrum and see the Eurobas that just discussed, the chimps, the Melanogaster, some bacteria, some wasp, some butterfly, some penguins, and except for the turtle, they seem to have like a U-shaped kind of spectrum, which means an excess of high frequency variant and low frequency variant, and that seems to be true for many different species. So visually is interesting, but we can use the statistical machinery. So what we've designed is a statistical machinery based on 3 different models. So the standard neutral model, the Kingman coalescent, with demography, with exponential growth and the orientation errors. A first model of multiple merger that's the beta coalescent, and the second one that's the psi coalescent. So I was very keen of like having at least 2 different models of multiple mergers to see if the results were different or not, right? And we ask 2 simple questions. Are the MMC models having some multiple merger component? Does it improve the fit on a statistical ground, right? So we use like maximum likelihood and base factors to pick the model and see. Here we have the 45 species. For 12 species, the best model is the Kingman model. But be aware that because the Kingman model has a parameter, has only 2 parameters in that implementation, it will be favored by the parsimony argument. And the second important point is that the Kingman model is included in the other models. So provided you would choose the other models, you could still have retrieve the Kingman models by having the parameter to the correct value. But for 75% of the model, the support is in favor either to the beta or the psi, and this MMC line here is where we cannot decide whether H1 or H2 is better than H0. I mean, they're better, but we don't know which one is better. So 75% of the side frequency spectrum points to a statistical support for an MMC kind of genealogy. And the second question is like, okay, it's better, but is it good? So we've came up with some kind of ranking based on Kramer's V, which is based on like a chi-square goodness of fit test. And we've graded A to D, A being like a very good fit and D being a very poor fit. And if we only keep the ones that have good support for multiple merger, still 62% are in good support for a multiple merger. So I think we've been using that standard neutral model, Kingman model, for like 50 years. But looking at the data, we should clearly use another model with an extra parameter, which would be a multiple merger kind of model. So if we look at the estimates of alpha, the strength of multiple merging, it ranges from 1 to 2, which means all possible cases are there. And there are no simple rules, such as the vertebrates have a smaller alpha or larger alpha, the bacteria have more multiple merging. So there is no simple rules like that. Everything everywhere. See, the cola is here. Some other bacteria are very strongly multiple merging. The penguin is there. The fly is here. The humans are there. Did not find any correlation. So I plot here the alphas, but if I plot the psi, it's the same. So just closing on this first case. So it's interesting to notice that the multiple merger correlation model that were mostly used by mathematicians, they can be defined in a proper timescale so that we can combine them with demography, structure, and all the tools we have in population genetics. The parameter estimation is straightforward, right? You can use either least square or maximum likelihood. That's very easy, and it gives the same answer. And on the biological side, very importantly, the standard neutral model we've been using for 50 years does not explain genome-wide polymorphisms, even if we had demography, and even if we had structure. I did not present that today. But there is more variance into the descendants. So either there is a large recurrent fluctuation in population size, so like crashes of populations all the time, possibly. Maybe there is a sweepstake effect, like sometimes there is a Genghis Khan or random Genghis Khan unrelated to natural selection that have many descendants, or by inheritance of cultural traits or by some reasons. Or it could also be the case that there is a recurrent relative sweeps known as a genetic draft that could explain the data. Now I have a second part I can do in say 10-15 minutes. Is it too long, or shall I stop there? Or please help me, Julia.
No, you can go on.
Okay, I go on for 10-15 minutes, not more. Okay.
Sure.
Okay, great. So the second attack to the standard neutral model is this concept of what we call effective population size that has exploded my mind for decades, and I'm just recovering it. I mean, my brain from the effective population size confusion. Okay, so here is the deal. You have a model, a standard neutral model on the left side, and we say constant population size, strict Parmigian, no selection, put many things there. Under that model, the time scale at which the genetic drift will operate, which is the time scale at which the ancestry between individuals will occur, is capital N, which is the population size. And as we mentioned at the very beginning of the talk, if you take a metric of diversity, it will be proportional to N times mu. And if you compute the average pairwise difference between individuals, that's going to be in a haploid version of the right Fischer model, 2n mu. Okay? That's a model. And on the right side, you have a real population. Anything you're going to put in your model, say constant population size, strict Mormixian, no selection, whatever you're going to put that, equal number of male and females, etc., it's likely going to be wrong in the real population. But instead of changing the model, what we've been doing for many decades is to say, yes, but it doesn't matter. What is the effective population size? What would be the population size so that the standard neutral model is somehow not completely wrong? Okay? So to do that, we can, because we cannot count the effective population size, but we can estimate the population size. And we can estimate the population size from the ancestry between the individuals. That is the time scale. How many generations do I have to go back in time to find an ancestor between two individuals? And we're going to call that effective population size. Okay? So here, to estimate the effective population size, we can simply retrieve the number of pairwise differences between individuals within a species and divide it by the mutation rate. And we're going to retrieve the how many generations do I need to go back in time to find a common ancestor, and call that effective population size. Somehow, the population size is the expected time scale, provided that the standard neutral model is true. And effective population size is the observed time scale, right? That's the expected number of generations I have to go back in time, N, and E is the observed number, inferred number of generations I have to go back in time to find a common ancestor. And now you can start computing numbers. Humans, 10 to the 10 individual, ancestry, 10,000 individuals, 10,000 generations, sorry. Gorilla, 100,000 individuals, ancestry, 1,000 generations. Drosophila melanogaster, no idea about the population size, very likely extremely large. I'm talking about worldwide. Huh? Time scale, 1 million generations to find a common ancestor between two individuals. And you can keep on doing that. There are fun ones, like there is a Promacrocrinus kerguelensis, that's a crinoid living all around the Antarctica continent. We've been computing the effective population size of that species, and it's 10. As if there were only 10 individuals in the population, right? Or E. coli, 10 to the 8. The ancestry is 10 to the 8 generations. Population size is likely, I asked Olivier Tenaillon, he told me like 10 to the 25, 10 to the 26. Some very big numbers. And so the question is like, why this column of NE has nothing to do with that column of N? So why diversity, why ancestry does not scale with the population size? And that's exactly what Leontine pointed out in 1974. That's a recent review on the subject, where they collected all the pairwise diversity in many different species, graphically plotted them, so that they expect the pairwise diversity. And these organisms, the pairwise differences, the diversity is in 1 or 2 orders of magnitude, but the number of individuals within those species, they are like they're covering very large range of order of magnitudes. So they're completely uncorrelated. That's extremely disturbing. So the second very general prediction of the standard neutral model is completely wrong as well, right? So what are the ideas? So we could think about population structuration. So structure, saying like the individuals are not permictic. So surely, locally, this will decrease diversity. I mean, if you belong to a small village with inbred people, you're genetically closer to your neighbors, but at the worldwide level, I think it would, on the contrary, tend to inflate the difference between individuals living at the different place on Earth. So I don't think that's the correct answer for that. So demography is a possible explanation, but that would mean that, for example, the E. coli, they were 10 to the 8, about 10 to the 8, some million years ago. So is that possible? So now they're 10 to the 24, but they have to have crashes of populations that goes up to 10 to the 8, like, I don't know, 10 to the 15. So you divide the population by 10 to the 15, which is a possibility. But you know, when we think about the gorillas and the elephant, when we divide their population by 10, we are already very scared. So by 100, we are like obfuscated. So by 10 to the 15, that's a very, very large demographical deviation. Huh? So another idea that someone gave me is like when the population gets very, very large, it splits into different species. So that within a species, the diversity is always at a maximum level. That's a possibility. And another possibility I'd like to discuss, at least briefly now, is the effect of selection. And not selection per se, but the effect of selection through linkage. So I still think that selection is a rare event, in the sense that there will be some selected variant spread in the genomes and spread into the population, but they're quite rare. But they have not only an effect on the selected loci, the selected locus, but they also have an effect on all the neighboring loci that are physically linked to that selected variant. So they can have a negative effect that is called a background selection, or a positive effect. Say you have a variant, like in this model. So you have a selected locus, that's the red locus, and a neutral locus, okay, that is somewhere away from the selected locus. You have a right Fischer population here with 5 individuals, and times goes from top to down. And at some point, there is a beneficial mutation arising in that red locus. And because it's beneficial, assume it spreads among the population. So what it will bring along with it is that gray circle that is physically linked with this red variant. So the gray circle will also spread with the selected variant, even though it has no effect on fitness. So that's called the hitchhiking effect. So the neutral variant is hitchhiking on the selected variant. So it does work when the recombination to selection coefficient is not very large. So if you're very far away, so see, you have a selected variant here, and it spreads through the population. If you look at the genealogy at a neutral locus that is genetically linked to the selected locus, some individuals will coalesce that correspond to that event. See, this is all the gray events. They will coalesce to this one, but the white circle, they will have a different history, and that's those ones. So what if now, if you look at the process backward, you have this process where the two locus are together, and anytime you have a recombination, they have a different genealogy, exactly as I've explained at the beginning of the talk, where the recombination, they uncouple the genealogies, right? So all the red individuals, they'll come back to that red first beneficial mutations, but the gray ones, some of them will coalesce here, some of them will escape the sweep. So imagine that you don't have a single sweep, but you have like mutations, beneficial mutations appearing here and there, sometimes in the different part of the genomes. They will like propagate into the genomes pseudo-independently, and they will bring the rest of the genome. Together with their sweep. And interestingly, if you're very close to the selected locus, all your nodes in your tree will be dictated by the sweeps, right? Any sweep will coalesce some of nodes, and what you will do is recover like some kind of a multiple merger coalescence. So if you're at the selected locus, it's a radiation. If you're close to the selected locus, not so far, you have a multiple merger coalescence. But if you go further away from the selected locus, interestingly, what you do recover is, see, there are some sweeps that are so far from that neutral locus, they don't affect the neutral locus. So they have no effect. So the coalescent event, they do correspond to some selected events, but not all. And interestingly, if you're far enough from the selected locus, you recover a completely Kingman neutral tree. It's a neutral. You cannot make the difference between this tree and a neutral tree, except that the time scale is completely different. And the time scale will be completely shortened. So now I see it's time. So that's a simulation showing that this works, some math we did with my colleague Emmanuel. Here is kind of an interesting plot where you have the effective population size as a function of the population size depending on your distance to the selected locus, right? So if you're very far away from the selected locus, your effective population size will be equal to your population size. But if you're not so far from a selected locus at a given time point, I mean, that means at your time scale, selective event will happen and will likely affect you a bit. Then your time scale is completely reduced and your effective population size is not anymore linear with the population size. It's a power law of the population size with a power smaller than 1, right? Say square root of the population size. What are they telling the data? Just look at the birds. If you look at diversity and population size, there is probably a power law. And recently there has been a paper with pinnipeds, and again they pointed to a power law between the population size and the diversity, which is the effective population size. So just discussing this, I think the riddle of diversity can be recast as a timescale problem, right? And a point that I did not touch is like we did many simulations, forward and backward, and the math, they all match, and we are quite happy with the model. Importantly, what does selection? It alters diversity not only as a selected locus, but on a very large fraction of the genomes on mega basis per selected variants through linkage. The effect will depend on the distance. If you're medium distance, your genealogical tree will look like an MMC. Interestingly, the timescale of the multiple merger is also a power law. At far distance, you'll recover a Kingman tree, so it looks like a neutral tree, but the timescale is the power law, again, of the population size. So we all agree that sequences do change, that there is some diversity in the locus. What we are still after is the unknown cause of molecular evolution. So in neutral theory, diversity should scale with n, which is wrong. And NE is a concept that was proposed to explain diversity using the neutral theory. Somehow, that's a way to define it. It can be discussed. But I think link selection is a good way to solve that paradox. And maybe what we do have, for example, in humans, why the population size, the effective population is so small is because even though the number of humans exploded like up to 10 to the 10, because of the draft, because of the selection, the effective population size is not linear with population size. It's n to the power of something smaller than 1, which would be a small increase. And maybe what we see in the PSMC and all the methods like inferring the variations of NE are variations of NE. They are not variations of the population size. And maybe considering link selection for this is a good explanation. Okay, thank you very much.